Anatomy of flowering plants. Study of internal structure of plants is called anatomy. Plants have dash as the basic unit. Cells are organized into dash and in turn the tissues are organized into. Plants have cells as the basic unit. Cells are organized into dash and in turn the tissues are organized into organs. Monocots and dicots are also seen to be anatomically different. True or false? True. Group of cells having common origin and usually performing a common function are called tissues. Tissues are classified into dash main groups, namely dash and dash tissues based on whether the cells being formed are capable of dash or not. Tissues are classified into two main groups, namely meristematic and permanent tissues based on whether the cells being formed are capable of dividing or not. Growth in plants is largely restricted to specialized regions of active cell division called meristems. Plants have different kinds of dash which occur at the tips of roots and shoots and produce primary tissues are called. Plants have different kinds of meristems. The meristems which occur at the tips of roots and shoots and produce primary tissues are called apical meristems. The meristem which occur between mature tissues is known as intercalary meristem. Meristose means divided. Meristose means divided. Root apical meristem occupies the dash, while the shoot apical meristem occupies distant most region of the root apical meristem occupies the tip of root and while the shoot apical meristem occupies the distant most region of the stem axis. During the formation of leaves and elongation of stem, some cells left behind from shoot apical meristem constitute axillary bud. Both apical meristem and intercalary meristems are primary meristem because they appear early in life of a plant and contribute to the formation of primary plant body. Meristems that occur in the mature region of roots and shoots of many plants, particularly those that produce woody axis and appear later than primary meristem, is called secondary or lateral meristem. Following divisions of cells in both primary and as well as secondary meristem, the newly formed cells become and specialize and lose the ability to. Such cells are termed as following divisions of cells in both primary and as well as secondary meristem, the newly formed cells become structurally and functionally specialized and lose the ability to divide. Such cells are termed as mature or permanent cell, constitute the permanent tissues. Axillary buds, which are present in the axils of leaves, are capable of forming a branch or flower. Intercalary meristem occur in dash and regenerate parts removed by the grazing herbivore grasses. Cylindrical meristems are primary or secondary. Secondary or lateral meristems. The cells of the permanent tissues do not generally divide further. True or false? True. The various simple tissues in plants are parenchyma, collenchyma, and sclerenchyma. Fascicular or vascular cambium is example for vascular cambium is example for secondary or lateral meristem. Permanent tissues having many different types of cells are called complex tissues. Interfascicular cambium is example for primary or secondary. Secondary or lateral meristem. Interfascicular cambium is example for secondary or lateral meristem. Permanent tissues having all cells similar in structure and function are called simple tissue. Cork cambium is example for secondary or lateral meristem. During the formation of primary plant body, specific regions of the apical meristem produce dermal tissues, ground tissues, and vascular tissues. These are responsible for the producing the secondary tissues, secondary or lateral meristem. A simple tissue is made of many types of cells. False. Simple tissue which form the major component within organs. Parenchyma. Simple tissue whose cells are generally isodiametric. Parenchyma. The simple tissue which occurs in layers below the epidermis in most of the dicotyledonous plants. Collenchyma. The simple tissue which may be oval or spherical or polygonal and often contain chloroplast. Collenchyma. Simple tissue which contains spherical, oval, round, or polygonal or elongated in shape. Parenchyma. Simple tissue which consists of long, narrow cells with thick and lignified cell walls having a few or numerous pits. Sclerenchyma. Simple tissue which found either as a homogeneous layer or in patches. Collenchyma. On the basis of variation in form, structure, origin, and development, sclerenchyma may be either fiber or scleroids. Simple tissue which consists of cells which are much thickened at the corners due to a deposition of cellulose or hemicelluloses and pectin. Collenchymas. Simple tissue which intercellular spaces are absent. Collenchyma. Simple tissue which may be either be closely packed or have a small intercellular spaces. Parenchyma. Collenchymatous cells assimilate food when they contain chloroplast. Thick-walled, elongated and pointed cell which generally occurring in groups in various parts of the plant. Fibers of sclerenchyma. Spherical or oval or cylindrical, highly thickened dead cell with very narrow cavities. Scleroids of sclerenchyma. Tissues which are made of more than one type of cell and work together as a unit are called complex tissues. Simple tissue which perform various functions like photosynthesis, storage, and excretion. Parenchyma. Cells of this simple tissue usually dead without protoplast. Sclerenchyma. Simple tissue which provide mechanical support to the growing parts of the plant, such as young stem and petiole of a leaf. Collenchyma. Simple tissue which provide mechanical support to the organs. Sclerenchyma. These are commonly found in the fruit walls of nuts, pulp of fruits like guava, pear, and sapota, seed coats of legumes and leaves of tea. Scleroids of sclerenchyma. Tissues in xylem and phloem of plants. Complex tissues. Conducting tissue for water and minerals from roots to the stem and leaves. Xylem. Complex tissue which provide mechanical strength to the plant parts. Xylem. Xylem is composed of four different kinds of elements, namely tracheids, vessels, fibers, parenchyma, the parenchyma fiber, tracheids, vessels, xylem fibers, and xylem parenchyma. Dash lack vessels in their xylem, gymnosperms. Elongated tube-like cells with thick and lignified walls and tapering ends, tracheids of xylem. Main water transporting elements in flowering plants are tracheids and vessels. Long cylindrical tube-like structure made of many cells, cells with lignified walls and large central cavity, no protoplasm. Vessel of xylem. Vessel of xylem. Highly thickened walls and obliterated central lumen, septate or aseptate. Xylem of fibers. Living and thin walled, cell walls are made up of cellulose, store food material in the form of starch or fat and tannins. Xylem of parenchyma cells. Element of xylem which are dead or without protoplasm and inner parts of the cell walls have thickening which vary in form. Tracheids of xylem. Vessel members are cells of vessel. Vessel members are interconnected through perforations in their common walls. Presence of vessel is a characteristic feature of angiosperms. It transports food material usually from leaves to other parts of the plant. Phloem. The radial conduction of water takes place by the ray parenchymatous cells. Ray parenchymatous cells. Two types of primary xylem are. The two types of primary xylem are protoxylem and metaxylem. First formed primary xylem elements are called protoxylem. Later formed xylem elements are called metaxylem. In stems, the protoxylem lies towards and the metaxylem lies towards dash of the organ. This type of primary xylem is called. In stems, the protoxylem lies towards the center and the metaxylem lies towards the periphery, and this type of primary xylem is called endark. Then protoxylem, location of protoxylem in roots, metaxylem location, and such a xylem is called. In roots, the protoxylem lies towards periphery and metaxylem lies towards center, and such arrangement of primary xylem is called exarch. Phloem is composed of sieve tube element, companion cell, phloem parenchyma, phloem fibers in angiosperms. Phloem is composed of albuminous cells and sieve cells, lac sieve tubes and companion cells in gymnosperms. Long tube-like structures arranged longitudinally and are associated with the companion cells are sieve tube elements. The tissue which stores food material and other substances like resins, latex, and mucilage. Phloem parenchyma. End wall of sieve elements are perforated in a sieve-like manner to form sieve plates. Cell wall of this tissue composed of cellulose and has pits through which plasmodesmatal connection exists between the cells. Phloem parenchyma. A mature sieve element possesses A and a large but lacks A. A mature sieve element possesses a peripheral cytoplasm and a large vacuole but lacks a nucleus. The functions of sieve tubes are controlled by the nucleus of companion cells. Specialized parenchymatous cells which are closely associated with sieve tube elements are companion cells. The sieve tube Elements and companion cells are connected by dash present between their common longitudinal walls. Pit fields. Phloem parenchyma in monocotyledons, absent mostly. These are generally absent in the primary phloem but are found in the secondary phloem fibers. Cells help in maintaining the pressure gradient in the sieve tubes. Companion cells. It is made up of elongated, tapering, cylindrical cells which have dense cytoplasm and nucleus. Phloem parenchyma. These are much elongated, unbranched, and have pointed, needle-like apices. Phloem fibers. Phloem fibers or bast fibers are made up of sclerenchymatous cells. Okay, we have answer here. Cell wall of phloem fibers is quite thick. At maturity, phloem fibers lose their protoplasm and become dead. Phloem fibers of dash and dash and dash are used commercially. Jute, flax, and hemp. First formed primary phloem consists of narrow sieve tubes and is referred to as, and the later formed phloem has bigger sieve tubes and it is referred to as protophloem and metaploem. First formed primary phloem consists of narrow sieve tubes and is referred to as protophloem, and the later formed is metaploem. On the basis of their structure and location, there are three types of tissue system. These are epidermal and ground or fundamental and vascular or conducting. Tissue system which forms the outermost covering of the whole plant body and comprises epidermal cells, stomata, and the epidermal appendages like the trichomes and hair is epidermal tissue system. Next, all tissues except epidermis and vascular bundles constitute ground tissue. The system that consists of complex tissues, the phloem and xylem, the system that constitutes of complex tissues, phloem and xylem, is vascular tissue. It consists of simple tissues such as parenchyma, collenchyma, and sclerenchyma. Ground tissue. The outside of the dash is often covered with a waxy thick layer called the dash, which prevent the loss of water. The outside of the epidermis is often covered with a waxy thick layer called cuticle, which prevent the loss of water. Structure which regulate the process of transpiration and gaseous exchange. Stomata. Cuticle is absent in roots. God cells are dumbbell shaped in grasses. The outer walls of God cells, which are away from the stomatal pore, are thin, and the inner walls are away from the stomatal pore are thin, and the inner walls are towards the stomatal pore, and they are highly thickened. The God cells possess and regulate the opening and closing of stomata. plus. Sometimes a few epidermal cells, sometimes a few epidermal cells in the vicinity of the guard cell become specialized in their shape and size are known as subsidiary cells. Are known as subsidiary cells. Stomatal aperture. Guard cells and the surrounding subsidiary cells are together called stomatal apparatus. Stomatal apparatus. Structures which are present in the epidermis of leaves are structures which are present in the epidermis of leaves are stomata. Outermost layer of the primary plant body is epidermis. Each stoma is composed of two dash-shaped cells, bean-shaped cells, known as guard cells. Parenchymatous cells of the ground tissues are usually present in cortex, pericycle, pith, and medullary rays in the primary stems and roots. In leaves, the ground tissue consists of thin-walled chloroplast containing cells and is called mesophyll. In dicotyledonous stems, cambium is present between, in dicotyledonous stems, cambium is present between phloem and xylem. It is made up of elongated, compactly arranged cells which form a continuous layer, and it is usually single layered. Identify this epidermal tissue, epidermis. If cambium, the xylem and phloem, the xylem and if cambium present in, wait, the xylem and phloem together constitute, the xylem and phloem together constitute vascular bundles. Cells of parenchymatous with small amount of cytoplasm lining the cell wall and a large vacuole. Identify these epidermal cells. Cells are parenchymatous with a small amount of cytoplasm lining the cell wall and a large vacuole is epidermis. If cambium present between the phloem and xylem, they possess the ability to form secondary xylem, and phloem tissues. Phloem tissues and hence are called open vascular bundles and hence are called open vascular bundle. Then it is possible in dicotyledonous stems. In the monocotyledons, the vascular bundles have no cambium present in them. Hence, since they do not form secondary, tissues. They are referred to as closed. When xylem and phloem within a vascular bundle are arranged in an alternate manner, along with the radii, vascular and alternate manner along with the different radii, the arrangement is called radial, such as in roots. In dash type of vascular bundles, conjoint type of vascular bundles. Xylem and phloem are jointly situated along the same radius of vascular bundles. Such vascular bundles are common in stems and leaves. The conjoint vascular bundles usually have the phloem located only on the outer side of xylem. In dicotyledonous roots, the outermost layer is outermost layer is epiblema. In dicotyledonous roots, several layers of thin-walled parenchyma cell with intercellular spaces are called cortex, and it is located below the epiblema. Monocotyledonous root. The anatomy of the monocot root is similar to the dicot root in many respects. In monocotyledonous root, the anatomy of the monocot root similar into the dicot root in many respects. True. In dicotyledonous roots, many of the cells dash protrude in the unicellular root hairs, epiblema. Monocot root has epidermis, cortex, endodermis, pericycle, vascular bundles. True. Pith, small and poorly developed in monocot root. False. Xylem bundles are few in dicot root and there are usually more than 6 polyarch xylem bundles in the monocot root. True. In dicotyledonous roots, the innermost layer of the cortex is called endodermis. In dicotyledonous roots, it contains a single layer of barrel-shaped cells without any intercellular spaces. Endodermis. In dicotyledonous roots, next to endodermis lies a few layers of thick-walled parenchymatous cells are present and referred to as pericycle. Initiation of lateral roots and vascular cambium during the secondary growth takes place in these cells. Parenchymatous cells of pericycle takes place in the parenchymatous cells of pericycle. In dicotyledonous roots, the tangential as well as radial walls of the endodermal cells have deposition of water impermeable waxy material, suberin, in the form of Casparian strips. Secondary growth in monocotyledonous roots. Secondary growth in monocotyledonous roots absent. The pith is small or inconspicuous in dicotyledonous roots. The parenchymatous cells of dicotyledonous roots, which lie between the xylem and phloem, are called conjunctive tissue. Conjunctive tissue. These are usually 2 to 4 xylem and phloem patches. There are usually 2 to 4 xylem and phloem patches. That is in dicotyledonous roots. Pith of monocot root is large, and well developed. A cambium ring develops between the xylem and phloem in dicotyledonous root. In dicotyledonous roots, all tissues on the inner side of the endodermis, such as pericycle, vascular bundle, pith, constitute. In dicotyledonous roots, all tissues on the inner side of the endodermis, such as pericycle, vascular bundles, and pith, constitute steel. Outermost protective layer of typical young dicotyledonous stem is epidermis. In dicotyledonous stem, the cells arranged in multiple layers between epidermis and pericycle constitute cortex. Epidermis covered with a thin layer of cuticle, and it may bear trichomes and a few stomata. Number of subzones in the cortex of dicotyledonous stem. 3. Outer subzone of cortex dicotyledonous stem, which consists a few layers of collenchymatous cells. Hypodermis. Subzone of cortex of dicotyledonous stem, which is present just below the epidermis. Hypodermis. Subzone of cortex of dicotyledonous, whose cells are rich in starch grains, and the layer is also referred to as the starch sheath. Endodermis. Subzone of cortex of dicotyledonous stem, which provide mechanical strength to the young stem. Hypodermis. Subzone of cortex of dicotyledonous stem, which is present just below the hypodermis, consists of rounded, thin-walled parenchymatous cells, with conspicuous intercellular spaces. The phloem parenchyma is absent. And water-containing cavities are present within the vascular bundles. Dicot stem or monocot stem? It's in monocot stem. Innermost subzone of cortex of dicotyledonous stem is called endodermis. The dash is present on the inner side of the endodermis and above the phloem in the form of semilunar patches of sclerenchyma. Pericycle. In dicot stem, each vascular bundle is conjoined, open, and with endark. And endark protoxylem. Vascular bundles of conjoined and closed in monocot stem. In dicotyledonous stem, in between the vascular bundles, there are a few layers of radially placed parenchymatous cells which constitute medullary rays. A large number of vascular bundles are arranged in a ring. A large number of vascular bundles arranged in a ring. The ring arrangement of vascular bundles is a characteristic feature of dicot stem. The monocot stem has a sclerenchymatous hypoderm. It has a sclerenchymatous hypoderm. A large number of scattered vascular bundles, each surrounded by sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. In dicot stem, a large number of rounded parenchymatous cells with large intercellular spaces which occupy the central portion of the stem constitute with, unfortunately we got answer here actually, peripheral vascular bundles are generally smaller than the centrally located one. Is it in dicot stem or monocot stem? In monocot stem. The vertical section of the dorsiventral leaf through the lamina shows three main parts, namely epidermis, mesophyll, and vascular system. Vascular system. The vertical section of the dorsiventral leaf through the lamina shows three main parts, epidermis, mesophyll, vascular system. Dorsiventral leaf, the vertical section of a dorsiventral leaf through the lamina shows three main parts, namely epidermis, mesophyll, and vascular system. Dicotyledonous leaf is called dorsiventral leaf. Dicotyledonous leaf is also called dorsiventral leaf. Monocotyledonous leaf is called isobilateral leaf. Anatomy of isobilateral leaf is similar to that of dorsiventral leaf in many ways. True or false? True. In dicotyledonous leaf, the epidermis which covers the upper surface is called adaxial epidermis. In dicotyledonous leaf, the epidermis which covers the lower surface is called abaxial epidermis. Type of epidermis which contain conspicuous cuticle in dicotyledonous leaf, both abaxial and adaxial. Type of epidermis which lack stomata, abaxial epidermis. Stomata are present on both the surfaces of epidermis in isobilateral leaf. In dicotyledonous leaf, the tissue between the upper and lower epidermis is called mesophyll. Mesophyll of dicotyledonous leaf possess dash and carry out photosynthesis. Chloroplast. Type of tissue present in the mesophyll of dicotyledonous leaf, parenchyma. The palisade parenchyma and spongy parenchyma are the unique feature of parenchyma of mesophyll of dicotyledonous leaf. Mesophyll is not differentiated into palisade and spongy parenchyma. Isobilateral leaf. Palisade parenchyma, location and shape of the cells. Location adaxially placed and shape of the cell, we can say elongated cells. Palisade parenchyma, arrangement of the cells, vertical and parallel to each other. Shape of cells of spongy parenchyma are oval or round. Arrangement of cells in spongy parenchyma, loosely arranged. Location of spongy parenchyma, situated below the palisade cells and extends to the lower epidermis. There are numerous large spaces and air cavities between these cells of parenchyma of dorsiventral leaf. Spongy parenchyma. Vascular system of dorsiventral leaf includes dash, which can be seen in the vascular system of dorsiventral leaf include vascular bundles and which can be seen in the veins and the mid-rib. In dorsiventral leaf, the size of the vascular bundle are dependent on the size of veins. The veins vary in thickness in the dash of the dicot leaves. The veins vary in the thickness of the reticulate venation of the dicot leaves. In dorsiventral leaf, the vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. Thick-walled bundle sheath cells. Certain adaxial epidermis cells, along with the veins, modify themselves into large, empty, colorless cells in dash, and they are called grasses. They are called bulliform cells. The parallel venation in dash leaves is reflected in the near similar sizes of vascular bundles except in as seen in vertical section of the leaf. The parallel venation in monocot leaves is reflected in the near similar sizes of vascular bundle except in main vein as seen in vertical sections of the leaves. If the bulliform cells in the leaves have absorbed water and are turgid, dash is exposed. Leaf surface is exposed. If the bulliform cells are flaccid, due to the water stress, they make dash curl inwards too. In bulliform cells, flaccid, due to the water stress, they make the leaves curl inwards to minimize water loss. The growth of the roots and the stems in length with the help of the growth of the roots and stems in length with the help of apical meristem is called primary growth. In dicot stem, the cells of cambium present between the primary xylem and primary phloem is the intrafascicular cambium. Apart from primary, if plants exhibit an increase in girth, it is called secondary growth. It is called secondary growth. The tissues involved in secondary growth are two lateral meristems, vascular cambium, and carp cambium. During secondary growth, vascular cambium is completely secondary in origin in dicot roots. Plants exhibit secondary growth mostly. Plant exhibit secondary growth mostly in dicotyledonous root plants. The meristematic layer that is responsible for cutting off vascular tissue xylem phloem is called vascular cambium. Excuse me. Vascular cambium. Vascular cambium originates from the tissue located just below the phloem bundles, a portion of pericycle tissue above the protoxylem, forming a complete and continuous wavy ring, which later becomes circular in dicot roots. Cells of medullary rays. Cells of medullary rays adjoining with intrafascicular cambium become meristematic and form the interfascicular cambium, thus a continuous ring of cambium formed in dicot stems. In the dash, vascular cambium is present in patches as a single layer between the xylem and phloem. Later, it forms a complete ring. It is possible in young stem. Interfascicular cambium of dicot root is formed by cells of medullary rays, intrafascicular cambium. Active cambial rim cut off new towards inner side or outer side, both sides. It will be on both sides. In active cambial ring cell, cut off towards the pith, mature into secondary xylem. In active cambial ring, cells cut off towards the periphery, mature into secondary phloem. Cambium generally more active on inner side or outer side? Inner side. Amount of secondary xylem produced is more than secondary phloem and soon forms a compact mass. Why? Cambium is generally more active on the inner side than the outer side. On the inner side than the outer side. That is the reason. The primary and secondary phloem get gradually crushed. Why? Due to the continued formation and accumulation of secondary xylem. The primary xylem remains more or less intact or in or around the center. Why? Due to the continued formation and accumulation of secondary xylem. So at some places forms a narrow band of parenchyma. At some places forms a narrow band of parenchyma which passes through the secondary xylem and the secondary phloem in the radial direction. These are called secondary medullary rays. The activity of cambium is under the control of many physiological and environmental factors. True or false? True. In temperate region, the climatic conditions are uniform throughout the year. True or false? False. In the dash season, cambium is very active and produce a large number of xylary elements having vessel with wider cavities. Spring. In which season the cambium is less active and form fewer xylary elements that have narrow vessels? Winter. Wood formed during spring season is called spring wood. Spring wood or early wood. Wood formed during winter season, autumn wood or late wood. Wood is lighter in color, lower density, spring wood. Wood is darker with higher density, autumn wood. Early wood formed in spring wood. Late wood is formed in autumn wood. Early wood formed in spring and it is called spring wood. Late wood is formed in autumn and it is called autumn wood. Xylary elements are more formed in spring. Xylary elements are less formed in autumn. The two kinds of woods that appear as concentric rings constitute an annual ring. By observing this, in cut stem we can estimate the age of tree by annual rings. Greater part of secondary xylem is dark, brown, hard, durable, and resistant to the microorganisms and insects. Comprises dead element, highly lignified walls. Such wood is called heartwood. Increase in girth of stem due to the activity of vascular cambium. Reason for the hardness durability of the heartwood is organic compounds like tannins, resins, oils, gums, aromatic substances, and essential oils in the central or innermost layers of the stem. Central or innermost layers of the stem. Wood which does not conduct water. Wood which does not conduct water. Heartwood or sapwood. Heartwood. Cork cambium is also called phellogen wood which gives mechanical support to the stem. Wood which gives mechanical support to the stem. Heartwood. Region which cork cambium developed in the stem. Region which cork cambium developed in the stem. Cortex. Wood comprises dead elements with highly lignified walls. Heartwood. It is a couple of layers thick. It is made of narrow, thin-walled, nearly rectangular cell. It cuts off cells on both sides. Phalogen or cork cambium, we can say. Bark that formed early in the season is called early bark. Also we call soft bark. Wood whose peripheral region of the secondary xylem is lighter in color. Sap wood. Wood which is involved in the conduction of water and minerals from the root and to leaf. Sap wood. As the stem continues to increase, wait a minute, we'll go down. As the stem continues to increase in girth due to the activity of vascular cambium, the outer, what do we call? Girth. Due to the activity of vascular cambium, outer girth and cortical layers get broken and need to be replaced to provide a new protective cell layer. Hence, sooner or later, another meristematic tissue called epidermis. Here, let me separate. Epidermis or cork cambium. Epidermis or cork cambium develops usually in the that carcemium, which is also called phalogen, in the cortex region. As the stem continues to increase in girth due to the activity of vascular cambium, the outer girth and cortical epidermis. Wait a minute, we have a small separation here. Cortical epidermis layers get broken and need to be replaced to provide new protective cell layer. And sooner or later, another meristematic tissue called carcambium or phalogen develops usually in the cortex region. So then outer cells of carcambium differentiate into carc or phellem. Differentiate into carc or phellem while the inner cells secondary differentiate into secondary cortex are also called phalloderm. Outer cells of the cork cambium differentiate into cork or phellem, while the inner cells differentiate into secondary cortex or phalloderm. The cork is impervious to water. Why? Due to suberin deposition in the cell wall. The cells of secondary cortex in cork cambium is parenchymatous. Bark formed at the end of the season is called late or hard bark. Tissue types in bark are periderm and secondary xylem. Secondary phloem. Tissues in bark, periderm and secondary phloem. Phalogen, phellem, phalloderm are collectively known as periderm. Due to the activity of the cork cambium, pressure builds upon the remaining layers and peripheral to phalogen, and ultimately these layers die and slag off. It is non-technically termed as bark. A non-technical term that refers to all tissues exterior to the vascular cambium, including secondary phloem, is bark. At certain regions of the cork cambium, the phalogen cuts off closely arranged parenchymatous cells on the outer side instead of cork cells. These dash soon. These parenchymatous cells soon rupture the epidermis, forming a lens-shaped opening called lenticels. Opening called lenticels. Here we have a small mistake in the end. We got the answer here. The lenticels is very important. That rupture the epidermis, forming a lens-shaped openings. We call them as lenticels. The last question here. Thanks for the patience. They permit the exchange of gases between the outer atmosphere and internal tissue of the stem. These occur in most woody trees. Lenticels. So, this is anatomy of flowering plants. Continuous revision is required as maximum flow I went through, and need to revise continuously. People need to revise continuously and try to read the textbook first, and then after that try to answer the questions before answer comes on the screen. And want to revise in future times, you people need to do mute and mute the video and make it in double speed. That makes you to revise properly for second time, third time when you are doing revision, that you have to do like this. Okay, so then thank you.